Good morning, all of you. I am Preeti from BSc Chemistry Honors 3rd year. Today we will read the chapter The Organic Mechanism of Coenzymes in Metabolism. Many enzymes cannot catalyze a reaction without the help of cofactor. Cofactor assist enzymes in catalyzing a variety of reactions that cannot be catalyzed solely by the amino acid side chains of protein. Some cofactors are metal ion while other are organic molecules. A metal ion cofactor act as a Lewis acid in a variety of ways to help an enzyme catalyze a reaction. It can coordinate with group on the enzyme, causing them to align in a geometry advantages for reaction. It can help bind the substrate to the active site of the enzyme. It can form a coordination complex with the substrate to increase its reactivity, or it can increase the nucleophilicity of water at the active site. An enzyme that has a tightly bound metal ion, that is CO2 positive, Cu2 positive, Fe2 positive, MO2 positive, Zn2 positive, is called a metalloenzyme. Carboxypeptidase A is an example of metalloenzyme. Cofactors that are organic molecules are called coenzymes. Coenzymes are derived from organic compounds commonly known as vitamins. A vitamin is a substance that the body cannot synthesize, that is needed in small amounts for normal body function. Sir Frederick Hopkins was the first to suggest that diseases such as rickets and scurvy might result from the absence of substances in the diet that are needed only in very small quantities. Because of first such compound recognized to be essential in the diet was an amine, Casimir Funkin incorrectly concluded that all such compounds were amines and all called them vitamins. The E was later dropped from the name. We have seen that enzymes catalyze reaction following the principle of organic chemistry. Coenzymes use these simple same principles. We will see that coenzymes play a variety of chemical roles that the amino acid side chains of enzymes cannot play. Some coenzymes function as oxidizing and reducing agent, some allow electrons to be delocalized, some inactivate groups for further reaction, and some provide good nucleophiles or strong bases needed for a reaction. Because it would be highly inefficient for the body to use a compound only once and then discard it. Coenzyme are recycled, therefore we will see that a coenzyme that is changed during the course of a reaction is subsequently converted back to its original form. An enzyme plus the cofactor it requires to catalyze a reaction is called a holoenzyme. An enzyme that has had its cofactor removed is called apoenzyme. Holoenzymes are catalytically active, whereas apoenzymes are catalytically inactive because they have lost their cofactors. Early nutritional studies divided vitamins into two classes: water soluble vitamins and water insoluble vitamins. Vitamin A, D, E, and K are water insoluble. Vitamin K is the only water insoluble vitamin currently known to function as a coenzyme. Vitamin A is required for proper vision. Vitamin D regulates calcium and phosphate metabolism, and vitamin E is an antioxidant because they do not function as coenzymes. Vitamin A, D, and E are not discussed in this chapter. Vitamin A and E are discussed in sections next chapter, and vitamin D is discussed in some other chapter. All the water soluble vitamins except vitamin C functions as coenzymes. In spite of its name, vitamin C is not actually a vitamin because it requires a fairly high amount and most mammals are able to synthesize it. Humans and guinea pigs cannot synthesize it. However, so it must include in their diets. We have seen that vitamin C and vitamin E are radical inhibitors and therefore are antioxidants. Vitamin C traps radicals formed in aqueous environments, whereas vitamin E traps radicals formed in non-polar environments. It is possible to overdose on water soluble vitamins because the body can readily eliminate any excess. You can, however, overdose on water insoluble vitamins because they are not easily eliminated by the body and can accumulate in cell membranes and other non-polar components of the body. For example, excess vitamin D causes calcification of soft tissues. The kidneys are particularly susceptible to calcification, which eventually leads to kidney failure. Vitamin D is formed in the skin as a result of photochemical reaction caused by ultraviolet rays from the sun. Overall view of metabolism. The reactions that living organisms carry out to obtain the energy they need and to synthesize the compounds they require are collectively known as metabolism. The process of metabolism can be divided into two parts, catabolism and anabolism. Catabolic reactions break down complex nutrient molecules to provide energy and simply precursor molecules for synthesis. Anabolic reactions require energy and result in the synthesis of complex biomolecules from simpler precursor molecules. And catabolism include complex molecules which will further give simple molecules plus energy. And anabolism includes simple molecules plus energy further give complex molecules. It is important to remember that almost every reaction that occur in a living system is catalyzed by an enzyme. The enzyme holds the reactants and any necessary cofactors in place so that the reacting functional groups and catalytic groups are oriented in a way that will cause a specific well defined chemical reaction. In some cases, it may be helpful to see where a particular enzyme catalyzed reaction discussed in this chapter fits into the overall metabolic scheme. So, we will start by taking an overall view of metabolism. Catabolism can be divided into four stages. The first stage of catabolism is called digestion. The reactants required for all life processes ultimately come from our diet. In that sense, we really are what we eat. In this first stage, fats, carbohydrates, and proteins are hydrolyzed into fatty acids, monosaccharides, and amino acids, respectively. In the second stage of catabolism, these hydrolysis products, fatty acids, monosaccharides, and amino acids, are converted to compounds that can enter the citric acid cycle. To enter the citric acid cycle, a compound must be either one of the compounds in the cycle itself, acetyl coenzyme A or pyruvate. Acetyl coenzyme A is the only non-citric acid cycle intermediate that can enter the cycle. Pyruvate can enter the citric acid cycle only because it can be converted to acetyl coenzyme A or to oxaloacetate. The citric acid cycle intermediate fatty acids are converted to acetyl coenzyme A, monosaccharides are converted to pyruvate via glycolysis, and amino acids are converted to acetyl coenzyme A, pyruvate, and or citric acid cycle intermediates depending on the amino acid. The third stage of catabolism is the citric acid cycle, also known as Krebs cycle or the tricarboxylic acid cycle. For every acetyl coenzyme A enters the cycle, two molecules of CO2 are formed. Metabolic energy is measured in terms of adenosine triphosphate. How the body uses ATP is described in next section. Very little ATP is formed in the first three stages of catabolism. However, in the fourth stage of catabolism, every NADH that is formed in the process of carrying out oxidation reactions in the earlier stages is converted into 3 ATPs in a process known as oxidative phosphorylation. Thus, the bulk of the energy provided by fats, carbohydrates, and proteins is obtained in this fourth stage. Anabolism can be thought of as the reverse of catabolism. In anabolism, acetyl coenzyme A, pyruvate and citric acid cycle intermediates are the starting materials for the synthesis of fatty acids, monosaccharides, and amino acids. These compounds are then used to form fats, carbohydrates, and proteins. The mechanisms utilized by biological systems to synthesize fats and proteins are discussed in sections. Next topic is niacin, the vitamin needed for many redox reactions. An enzyme that catalyzes an oxidation reaction or a reduction reaction requires a coenzyme because none of the amino acid side chains can act as an oxidizing or reducing agent. The coenzyme is the oxidizing or reducing agent. The enzyme's role is to hold the substrate and coenzyme together so that the oxidation or reduction reaction can take place. The coenzymes most commonly used by enzymes to catalyze oxidation reaction are nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate. When NAD positive oxidizes a substrate, the coenzyme is reduced to NADH. NADH and NADPH are reducing agents. They are used as coenzymes by enzymes that catalyze reduction reactions. Enzymes that catalyze oxidation reactions bind NAD positive more tightly than they bind NADH. When the oxidation reaction is over, the relatively loose bound NADH dissociates from the enzyme. Likewise, enzymes that catalyze reduction reactions bind NADH more tightly than they bind NAD positive. When the reduction reaction is over, the relatively loosely bound NAD positive dissociates from the enzyme. NAD positive is composed of two nucleotides linked together through their phosphate groups. A nucleotide consists of a heterocycle attached in a beta configuration to C1 of the phosphorylated ribose. A heterocyclic is a cycle compound in which one or more of the ring atoms in an atom other than carbon. The heterocyclic component of one of the nucleotides of NAD positive is nicotinamide, and the heterocyclic component of other is adenine. This accounts for the coenzyme's name. The positive charge in the NAD positive abbreviation indicates the positively charged nitrogen of a substituted pyridine ring. The only way in which NAD positive and differs structurally from NAD positive is in the phosphate group bonded to secondary OH group of ribose of adenine nucleotide. This explains the addition of P to the name NAD positive, and NADH are generally used as coenzymes in catabolic reactions. NADP positive and NADPH are generally used as coenzymes in anabolic reactions. The adenine nucleotide for the coenzymes is provided by ATP. The nicotinamide nucleotide is not synthesized by biological systems but is derived instead from the vitamin known as niacin. The oxidation of the secondary alcohol group of malate to a ketone group is one of the reactions in citric acid cycle. NAD positive is the oxidizing reagent in this reaction. Many enzymes that catalyze oxidation reactions are called dehydrogenases. Recall that the number of CH bonds decreases in an oxidation reaction. In other words, dehydrogenases remove hydrogen. Beta aspartate semi-aldehyde is reduced to homoserine in an anabolic pathway with NADPH as the reducing agent. The differentiation between the coenzymes used in catabolism and anabolism is maintained because the enzymes that catalyze these oxidation reduction reactions exhibit strong specificity for a particular coenzyme. For example, an enzyme that catalyzes Oxidation reaction can readily tell the difference between NAD positive and NADP positive. If the enzyme is in a catabolic pathway, it will bind NAD positive but not NADP positive. In addition, the relative concentrations of the coenzyme in the cell encourage binding of appropriate coenzyme. For example, because NAD positive and NADH are catabolic coenzymes and catabolic reactions are most often oxidation reactions, the NAD positive concentration in the cell is much greater than NADH concentration. Because NADP positive and NADPH are anabolic coenzymes and anabolic pathway are predominantly reduction reactions, the concentration of NADPH in the cell is greater than the concentration of NADP positive. Mechanism for pyridine nucleotide coenzymes. How do these oxidation reduction reaction takes place? All the chemistry of the pyridine nucleotide coenzymes takes place at the 4 position of pyridine ring. The rest of the molecule is important for the binding of the coenzyme to proper site of the enzyme. If a substrate is being oxidized, it donates a hydride ion to 4 position of pyridine ring. In the following reaction, the primary alcohol is oxidized to an aldehyde. A basic amino acid side chain of the enzyme can help the reaction by removing a proton from the oxygen in the substrate. Glyceraldehyde 3-phosphate dehydrogenase is an example of an enzyme that uses NAD positive as an oxidizing coenzyme. The enzyme catalyzes the oxidation of the aldehyde group of glyceraldehyde 3-phosphate to an anhydride of a carboxylic acid and phosphoric acid. This is a reaction that occurs in glycolysis. In the first step of mechanism for this reaction, an SH group of a cysteine side chain at the active site of the enzyme react with glyceraldehyde 3-phosphate to form a tetrahedral intermediate. A side chain of the enzyme increases cysteine's nucleophilicity by acting as a general base catalyst. The tetrahedral intermediate expels a hydride ion, transferring it to the 4 position of pyridine ring of an NAD positive that is bonded to the enzyme at an adjacent site. NADH dissociates from the enzyme and the enzyme binds a new NAD positive phosphate react with the thioester, forming the anhydride product and releasing cysteine. Notice that at the end of the reaction, the hollow enzyme is exactly as it was at the beginning, so the catalytic cycle can be repeated. The NADH produced in the reaction is reoxidized to NAD positive in the fourth stage of catabolism. The mechanism for reduction by NADH is the reverse of the mechanism of oxidation by NAD positive. If a substrate is being reduced, the dihydropyridine ring donates a hydride ion from its fourth position to the substrate. An acidic side chain of the enzyme aids the reaction by donating a proton to the substrate. Because NADH and NADPH reduce compounds by donating a hydride ion, they can be considered biological equivalents of sodium borohydride and lithium aluminum hydride, donors used as reducing reagents in non-biological reactions. Why are the structure of biological redox reagents so much more complicated than the structure of redox reagents used to carry out the same reaction in the laboratory? NADH is certainly more complicated than lithium aluminum hydride, although both reagents reduce compounds by donating a hydride ion. Much of the structural complexity of a coenzyme is for molecular recognition to allow it to be recognized by the enzyme. Molecular recognition allows the enzyme to bind the substrate and the coenzyme in the proper orientation for reaction. In addition, a redox reagent found in a biological system must be less reactive than a laboratory redox reagent because it must be selective For example, a biological reducing agent cannot reduce just any reducible compound with which it comes into contact. Biological reactions are much more carefully controlled than that because the coenzymes are relatively unreactive compared with non-biological redox reagents. The reaction between the substrate and the coenzyme does not occur at all at or take place very slowly without the enzyme. For example, NADH will reduce an aldehyde or a ketone only in the presence of an enzyme. Sodium borohydride and lithium aluminum hydride are more reactive hydride donors, much too reactive to exist in the aqueous environment of the cell. Similarly, NAD positive is a much more selective oxidizing agent than the typical oxidizing agent used in laboratory. For example, NAD positive will oxidize an alcohol only in the presence of an enzyme because a biological reducing agent must be recycled rather than having its oxidized form thrown away, as would be the case for a reducing agent used in laboratory. The equilibrium constant between the oxidized and reduced forms is generally close to unity. Therefore, biological redox reactions are not highly exergonic, rather, they are equilibrium reactions that are driven in the appropriate direction by the removal of reaction products as a result of participation in subsequent reactions. As you study the coenzymes in this chapter, don't let the complexity of their structure deter you. Notice that only a small part of coenzyme is actually involved in the chemical reaction. Notice also that coenzymes follow the same rules of organic chemistry as do the simple organic molecules with which you are familiar. We saw that an oxidizing enzyme can distinguish between the two hydrogens on the carbon from which it catalyzes the removal of a hydride ion. For example, alcohol hydrogenase removes only the pro-R hydrogen HA of ethanol, HB in pro-S hydrogen. Similarly, a reducing enzyme can distinguish between the two hydrogens at fourth position of the nicotinamide ring of NADH. An enzyme has a specific binding site for the coenzyme, and when it binds the coenzyme, it blocks one of the sites. If the enzyme blocks the B side of NADH, the substrate will bind to A side and HA hydride ion will be transferred to the substrate. If the enzyme blocks the A side of the coenzyme, the substrate will have to bind at the B side and the HB hydride ion will transfer. Currently, 156 hydrogenases are known to transfer HA and 121 are known to transfer HB. Flavin adenine dinucleotide and flavin mononucleotide, that is vitamin B2. A flavoprotein is an enzyme that contains flavin adenine dinucleotide or flavin mononucleotide. As an coenzyme, FAD and FMN like NAD positive and NADP positive are coenzymes used in oxidation reactions. As its name indicates, FAD is a dinucleotide in which one of the heterocyclic component is flavin and other is adenine. FMN contains flavin but not adenine. It is a mononucleotide. Notice that instead of ribose, the flavin nucleotide has a ribotyl group. Flavin plus ribotyl is called riboflavin. Riboflavin is also known as vitamin B2. A vitamin B2 deficiency causes inflammation of the skin. In most flavoproteins, FAD is bound quite tightly. Tight binding allows the enzyme to control the oxidation potential of the coenzyme. Consequently, some flavoproteins are stronger oxidizing agents than others. How can we tell which enzyme use FAD rather than NAD positive? A rough guideline is that NAD positive and NADP positive are the coenzymes used in enzyme catalyzed oxidation reactions involving carbonyl compounds, while FAD and FMN are the coenzymes used in other types of oxidations. For example, in the following reactions, FAD oxidizes a dithiol to a disulfide, an amine to an imine, and a saturated alkyl group to an unsaturated alkene, and FMN oxidizes NADH to NAD positive, an approximate guideline. However, because FAD is involved in some oxidations that involve carbonyl compounds, and NAD positive and NADP positive are involved in some oxidations that do not involve carbonyl compounds. Mechanism for flavin nucleotide coenzymes. When FAD oxidizes a substrate, the coenzyme is reduced to FADH2. FADH2 and FMNH2 are like NADH and NADPH are reducing agents. All the oxidation reduction chemistry takes place on the flavin ring. Reduction of the flavin ring disrupts the conjugated system, so the reduced coenzyme are less colored than their oxidized forms. In the first step of mechanism of FAD, catalyzed oxidation of dihydropoate to lipoate, the thiolate ion attacks the C4A position of flavin ring. This reaction is general acid catalyzed. As thiolate ion attacks the ring, a proton is donated to N5 nitrogen. A second nucleophilic attack by a thiolate ion, this time on the sulfur that is covalently attached to the coenzyme, generates the oxidized product and FADH2. In the first step of flaming catalyzed oxidation of amino acid to an amino acid, a basic side chain at the active site of enzyme removes a proton from alpha carbon of amino acid. The carboanion that is formed attacks the N5 position of flavin ring. Collapse of the resulting tetrahedral intermediate gives the oxidized amino acid and reduced coenzyme FADH2. Notice that FAD is more versatile coenzyme than NAD positive. Unlike NAD positive, which always use the same mechanism, flavin coenzymes can use several different mechanisms to carry out an oxidation. For example, we have just seen when FAD oxidizes dihydropoate, nucleophilic attack occur at the C4A position of the flavin ring. But when it oxidizes an amino acid, nucleophilic attack occur on N5 position. Cells contain very low concentration of FAD, much higher concentration of NAD positive. This difference in the concentration is responsible for a significant difference in the enzymes that use NAD positive as an oxidizing agent and those that use FAD generally. FAD is covalently bound to its enzyme and remain bound after reduced to FADH2. Unlike NADH, FADH2 does not dissociate from the enzyme. It therefore must be reoxidized to FAD before the enzyme can enter round of the catalysis. The oxidizing agent used for this reaction is NAD positive or O2. Therefore, an enzyme that uses an oxidizing coenzyme other than NAD positive may still require NAD positive to reoxidize the reduced coenzyme. For this reason, NAD positive has also been called common currency of biological oxidation reduction reactions. Thiamine pyrophosphate, vitamin B1. Thiamine was the first of the B vitamins to be identified, so it became known as vitamin B1. The absence of thiamine in the diet causes a disease called beriberi, which damages the heart and impairs nerve reflexes. In extreme cases, it causes paralysis. One major dietary source of vitamin B1 is hulls of rice kernels. A deficiency is therefore most likely to occur when highly polished rice is a major component of the diet. The deficiency is also seen in alcoholics who are severely malnourished. The coenzyme form of vitamin B1 is thiamine pyrophosphate. TPP is the coenzyme required by the enzymes that catalyze the transfer of two carbon fragments from one species to another. Pyruvate decarboxylase, an enzyme that requires thiamine pyrophosphate. Pyruvate decarboxylate catalyzes the decarboxylation of pyruvate and transfer the resulting 2 carbon fragment to a proton, resulting in the formation of acetaldehyde. You may wonder when alpha keto acid such as pyruvate can be decarboxylated, since the electrons left behind when CO2 is removed cannot be delocalized onto the carbonyl oxygen. We will see that thiazolium ring of coenzyme provide a site for the delocalization of electrons. A site to which electrons can be delocalized is called electron sink. Mechanism for pyruvate decarboxylase. The hydrogen bonded to imine carbon of TPP is relatively acidic because the allyl carbanion formed when the proton is removed is stabilized by adjacent positively charged nitrogen. The allyl carbanion is a good nucleophile. In the first step of the mechanism for pyruvate decarboxylase, the nucleophilic carbanion attacks the electrophilic ketone group of a alpha keto acid. The resulting intermediate can easily undergo decarboxylation because the electrons left behind when CO2 is removed can be delocalized onto the positively charged nitrogen. The positively charged nitrogen of TPP is a more effective electron sink than the beta keto group of beta keto acid, a class of compounds that are fairly easily decarboxylated. The product of decarboxylation is stabilized by electron delocalization. One of the resonance contributors is neutral and the other has separated charges. We will call it a resonance stabilized carbanion. Protonation of the resonance stabilized carbanion and a subsequent elimination reaction from form acetaldehyde and regenerate the coenzyme. Mechanism for pyruvate dehydrogenase system. A compound must enter the citric acid cycle to be completely metabolized. Those fats, carbohydrates, and proteins must be converted into the compound that are part of citric acid cycle that can enter the cycle. Acetyl coenzyme A is the only compound capable of entering the citric acid cycle. The final product of carbohydrate metabolism is pyruvate. To enter the carboxylic acid cycle, pyruvate must be converted either to acetyl coenzyme A or to oxaloacetate, a citric acid cycle intermediate. The pyruvate dehydrogenase system is a group of 3 enzymes responsible for the conversion of pyruvate to acetyl coenzyme A. The pyruvate dehydrogenase system requires TPP and other 4 coenzymes, that is lipoate, coenzyme A, FAD and NAD positive. The first enzyme in the system catalyzes the reaction of TPP with pyruvate to form the same resonance stabilized carbanion formed by pyruvate decarboxylase, and by the enzyme, the second enzyme of the system requires lipoate, a coenzyme that becomes attached to its enzyme by forming an amide with the amino group of lysine side chain. The disulfide linkage of lipoate is cleaved when it undergoes nucleophilic attack by the carbanion. In the next step, the TPP carbanion is eliminated from tetrahedral intermediate. Coenzyme A reacts with the thioester in a trans-thioesterification reaction. One thioester is converted into another, substituting coenzyme A for dihydropolate. At this point, the final reaction product has been formed. However, before another catalytic cycle can occur, dihydropolate must be oxidized back to lipoate. This is done by the third enzyme, E3 and FAD requiring enzyme. Oxidation of dihydrolipoate by FAD forms enzyme bound FADH2, NAD positive, then oxidized FADH2 back to FAD. The vitamin precursor of coenzyme ASH is pantothenate. We have seen that coenzyme ASH is used in biological systems to activate carboxylic acids by converting them into thioesters, which are much more reactive towards nucleophilic acyl substitution reactions that are carboxylic acids at physiological pH 7.3. A carboxylic acid would be present in its negatively charged basic form, which could not be approached by a nucleophile. Thank you.